Hello, my name's Drew Hartley, and I worked with Sam McChesney on this research presentation. Together, we looked at FMRP and its role in regulating neurogenesis in vivo in Xenopus laevis tadpoles.
Let's first discuss the topic of neurogenesis. It is defined as the process by which new neurons are formed in the brain. It plays a major role in helping stem cells mature into fully functioning neurons and is crucial during the development of the embryo.
Fragile X syndrome, or FXS, is the most common monogenetic form of autism, and it results from the silencing of the FMR1 gene during embryonic development. The FMR1 gene is responsible for producing Fragile X mental retardation protein, or FMRP, which is an RNA binding protein that regulates mRNA transport, stability, and translation. Developing embryos with FXS lose functionality of FMRP, which leads to intellectual disability. While FXS is known to be a disease that's caused by synaptic dysfunction, it's not known how FMRP contributes to the development of this condition. Synaptogenesis, which is the generation of new synapses, occurs well after FMRP expression is lost, so the relationship between these variables has yet to be determined. Displayed below are the materials and methods used within this experiment, the most important of which was in vivo time-lapse imaging of these labeled cells. This type of imaging allowed researchers to track the growth of the cells as a function of time, something that previously has not been done in any experiments before.
After compiling everything in the experiment, they found many things to be true in the tadpoles. The first thing was that low FMR1A MOs, which were 0.05 micrometers, created very variable knockouts, which implies that the threshold for detection lies just within the 0.05 micrometers. Next was that in the in vitro assay, meaning the experiment in the in vitro tadpole, the results showed MOs leading to decreased translation and transcription, which lowered the overall expression of the protein of interest. Then they found that just overall FM1A MOs are sufficient means to block and knock out FMRPs in cells and their expression. One important thing to note that they found was that different levels of MOs meant different levels of knockdown, meaning FMRP is not all or nothing when it comes to blocking it, but instead can be variable in expression. Another result that they found was that the delta FMR1 T2A EGFP can be used to overexpress FMRP, which can normally lead to inhibition or too much stimulation in the next protein of the cascade. They also found that FMRP-OE can be used to test sensitivity of neurogenesis, and the MFRP decreases cell proliferation as well as cell survival in vitro. Finally, they found that overexpression of FMRP, most specifically in the optic tensum of the tadpole, affects cell proliferation and cell survival in the brain, and that high concentrations of MO leads to increased cell death due to a knockdown of FMRP. Tadpoles were chosen for the experiment because by using MOs to block translation of FMRP and induce FXS, they were able to recreate the loss of FMRP in human fetal development. More importantly, in humans, FMRP lasts for 12.5 weeks in vitro with FSX-related fetuses, which is important because FSX-affected individuals normally don't have any FMRP, meaning that finding a way to recreate the fetus having FMRP for a set amount of time is very important. By using tadpoles, they were able to not wait to knock out FMRP in vitro until it was safe to assume it would be at the same stage that a human fetus would be during the FSX progression. Tadpoles were also good because FMRP in humans is found in the stem cells before differentiation, which tadpoles have initially, and is good at recreating the human model before knockouts by MOs occurred. Finally, through MOs, they were able to knock out the FMRP to recreate the loss of FMRP in FSX individuals. In the experiment, tadpoles were tested by knocking down FMRP, meaning that FMRP is made inoperative and can no longer be expressed. Through electroporating MOs, In high knockout cells, the tadpoles showed large decreases in cell proliferation, which is the process of increasing the amount of cells overall while overcoming cell death, and cell survival, meaning large amounts of apoptosis occurs. In order to find out that cell proliferation and cell survival decrease with high FMRP knockout, they use CLDU, which is a thymidine analog, which are used to study the miscoding potential of DNA precursors. And they also use cytox staining, which is a stain that only colors cells with weakened membranes. And because the cells were electroporated with MO, the cell membranes are weakened in order to knock out the FMRP, meaning the stain will only color the cells of interest containing the MOs and the NPCs. High knockout of FMRP is also related to NPC differentiation, which ends up leading to a simpler dendritic arbors. This overall leads to a small cell population, even though there are more neurons overall. Next, tadpoles were also tested with low FMRP knockdown. They found that it really affects the NPC apoptosis by increasing it with lower amounts of MO used. This ultimately leads to increased NPC cell death. Even though all of this occurs, the actual amount of NPCs in neurons doesn't change, meaning low FMRP knockdown is normal in differentiation of the cells. Low MFRP knockdown also creates poor dendritic arbor development, which makes sense because high knockdown in the cell leads to increases of NPCs and simple dendritic arbors made. In the next portion, they discuss low level of FMRP overexpression. In low levels of overexpression, there are no major defects to cell proliferation, differentiation, and dendritic morphology, meaning that dendrites contain the correct shape and therefore the correct pathways to express everything correctly. This shows that these cells in tadpoles have a basic tolerance for low levels of overexpression and can combat the slight difference in overexpression. Then, in high levels of FMRP overexpression, the over the overexpression reduces the overall increase of cell numbers during the first 3 days of development. This means that NPC proliferation is decreased in NPC cell survival. The overexpression of FMRPs, though in terms of differentiation, is way more important to the cells than the high knockout of FMRPs. Then, just like high levels of knockdown, the high level of overexpression also have simple dendritic arbors. From all of this, then they studied the difference in cell proliferation. In a decrease of cell proliferation, they found that it was caused by overexpression or loss of FMRP in the tadpole's optic tentum. This overall leads to FSX occurring in the individual, which displays microcephaly, dilation of ventricles, cortical atrophy, and dendritic spine oddities. All of this occurs because regions of the brain are all changed in size because of the difference in cell proliferation and therefore cell quantity, which ultimately affects the brain's ability to function properly.
Results of this experiment show that FMRP functions cell autonomously to regulate cell proliferation, survival, and dendritic morphology. Time-lapse imaging allowed experimenters to observe the growth slash maturation of these NPCs and led to the new postulation that cell survival and proliferation may be less sensitive to FMRP levels than neuronal differentiation. These results also show that decreasing or increasing FMRP expression increases apoptosis, meaning that proper development of neurons is highly regulated by FMRP. Lastly, while it was determined that FMRP levels heavily influence the growth of dendritic arbors in developing brains, no research has been conducted regarding the effects of varying FMRP levels within matured brains. Although there have been previous studies regarding the effect of FMRP on neuronal development, many of the results have been ambiguous. The use of in vivo time-lapse imaging in this experiment gives one a much finer means of measurement because it allows the researchers to resolve changes over time in this growth. In applying these findings to clinical practices, most FXS research currently focuses on glutamatergic synaptic signaling. Conversely, this study suggests that disorders such as this one may result from developmental errors that occurred prenatally. Therefore, this new theory on development of disease offers new doors to possible therapeutic methods of benefit. Although this experiment included many new means of observation, there is always room for experimental improvement. In furthering this study, one could look into the longer-term effects of FMRP on cell development, rather than the 3-day study conducted in this one. In addition, the use of a stain that would allow experimenters to identify cells that were previously marked as unidentifiable would be very beneficial. This would allow one to take a closer look at the effect of FMRP on the differentiation of cells. Overall, this study was conducted very effectively, and the detail allowed for a great analysis of the data.